Good evening. 10:30 p.m. my time anyway. Um, I'm making a second video in this short series. Maybe it'll become long, sorry, if I'm lucky. To share some thoughts I've been having the last few days on how the melanin and the idea that glutamate polymers exist in the brain and that they are analogous to chlorophylls and chloroplasts of plants, because it sort of makes a lot of things kind of fall out. And I'm going to record this before I get struck by lightning or something like that. And on my mind, it's easier for me to just blurt them out, and I'll try to do this in a whiteboard fashion. And almost certainly I've got a lot of details wrong, but almost certainly I've also got a lot of the core details right, and that it has a lot of implications for medicine and understanding biochemistry in life. So I'm going to maybe start with a whiteboard and start drawing some things and just start talking about, I'll try to make this sort of my elevator talk, if you will, on, what I think is going on. So this should be bringing up a whiteboard. I'm going to draw on that. And let's see, I can make that more narrow. Okay, I'll try 3 point. So I'm going to kind of start by maybe sketching out some of the relevant stuff. And if you watch that previous video, you'll already sort of have an idea what's going on. I know it was pretty long and hardcore, and probably a lot of my friends and people that aren't quite as involved in the ideas struggle very much with following my line of thought in some places. So this is what I'm sketching out, and I'm hoping this will record adequately. Excuse me, just had a dinner, trying to get some dinner before I recorded this. So I'm hoping this will, at least the audio was good last time, and we're hoping the video this time won't drive us batty, which it was doing last time. And I don't think I even used the whiteboard last time, and so we'll give this a shot. Okay, so I'm gonna sketch out. Whoops. All right, well, this will work. All right, this is L-dopa. This is the drug they give to patients that are suffering from Parkinson's disease. So I'll start with that sort of introduction as far as why do we give patients Parkinson's? Why do we give Parkinson's patients L-dopa? And so the core idea is that there's a region of the brain called the substantia nigra, and it's got this name because it's substance black substance is really what the term means. And it was discovered over 300 years ago when Parkinson's patients' brains were examined under the microscope, and you could tell very clearly that they were less pigmented in comparison to normal counterparts. And so that's now understood that the loss of that pigmentation is associated with Parkinson's disease. And so my hypothesis has been that the melanin acts as a dopamine storage and a memory system, effectively. And so I want to build on that and think about how some of the mechanism of that. Try not to, again, get out of hand. And so I'll have to redraw stuff, but the vision of a dopaminergic neuron is that it packages and stores dopamine in these vesicles that are near a junction between two neurons, and so we call this the synapse. And I went over this in detail the other time, but just briefly, effectively, it's thought that an electrical signal travels down the length of the membrane through a depolarization event, an ion flow across the membrane. So a lot of things could trigger that injury, or light at the retina of the eye, for example. And a lot of that's not well understood as far as what signals can get here and how, and that's part of my understanding now, is that photons are probably getting to the brain along a pathway of pi conjugated lipids in the membrane fraction. But to get to that, it takes a lot of explanation, and again, I'm trying to make this elevator talk for Parkinson's and melanin, and then now trying to extend that actually to glutamate polymers and what that has to do with Alzheimer's disease. So when I was taught this process, I understood that it involves calcium ions, and that the calcium flow into the neuron to interact with the lipid, phospholipid environment here has a lot to do with that depolarization event. As the signal comes down, calcium enters the cell and interacts with the lipid fraction around the vesicles, in particular with the phosphate, and making a bridge of sorts, which leads to either vesicle formation or dissolution. It sort of depends on how it's from where it's being used, but in this case, causing the dopaminergic vesicle to fuse with the plasma membrane, and so dopamine is released into the cytoplasm, and that this is enough to allow the electric signal to jump that gap effectively. And there's a lot of different ways you can think about that. If you take L-DOPA, it is something, well, first of all, you can't just give dopamine to a patient and restore their dopamine levels in the brain. And here's why. I've circled the carboxylic acid group of L-DOPA, the drug that they actually give Parkinson's patients. And what is true is that this carboxylic acid group is one of the easiest things to remove to gain energy if you're like a bacterium or anything else. And a lot of the things we eat actually are amino acid. And this is an example of an amino acid, but L-DOPA is an amino acid that has had a hydroxyl group added to it. And so, in other words, and it's a little bit hard to picture now, but this ring here, or the circle, that's an aromatic ring. It has some magnetic properties associated with it that are important here. There's a single OH group, a hydroxyl group, we call it, on the amino acid tyrosine, and the carboxylic acid group here and the amine are both on that amino acid tyrosine. And so if you ate protein containing foods and they were hydrolyzed in your stomach, this is one of the things that presumably would get to your brain very readily. And part of the reason it does that, and the reason why dopamine itself doesn't do it, is because it's something we call a Zwitter ion. That is, if you look at the structure I've drawn here of L-DOPA, and L-DOPA has everything, including the circled stuff here on this figure. But let me change colors. What's true is that one of the hydrogens that's part of this structure, if you actually drew it in three-dimensional space, it would look very different than the way I've drawn it here, which is something of an illusion. That is, there's a hydrogen, a proton that's in a bridge sort of state that allows this amino acid that has both an amine and a carboxylic acid on it. It allows it to either be charged and interact with water fraction, because it can adopt the configuration that I drew, or it can, the proton can just sort of blip and be on the carboxylic acid and protonate it, and that essentially makes the amine neutral, and so they're both neutralized. And that thing is now a neutral hydrophobic molecule, and it goes straight through membranes. So L-DOPA and things like dopamine itself get into the dopaminergic neurons. The amino acid form will get to the brain past the blood-brain barrier because of this Witterite character. Once it's in the brain, in the case of L-DOPA, it's taken up into the neuron, and it's quickly converted to dopamine by removal of that carboxylic acid group, and through now at least the enzyme aromatic amino acid decarboxylase. But this is a reaction that's would happen readily anyways. It's almost, it's fairly spontaneous. It's definitely a spontaneous reaction, or it wouldn't really occur. But, well, it could with the addition of ATP. But so but when it enters the cell, then though it's in this form, and so there's essentially a CO2 that's gone away, and so this dopamine then is packaged up into vesicles, presumably, and then enables a patient to release that dopamine to the synapse and restore the dopamine that they were not otherwise getting. And so the question is, why weren't they getting that dopamine? And the normal pathway for making dopamine is thought to involve starting with the amino acid, which is everything not circled in red on this structure here. That is tyrosine. The amino acid is hydroxylated through tyrosine hydroxylase, an enzyme that's prominent in dopaminergic neurons of the brain. And this is the enzyme that's used as an immunologic marker for histologic staining of dopaminergic neurons in brain tissue to show this is a dopaminergic neuron because it has tyrosine hydroxylase in it. And importantly, it was thought for a long time that those cells decreased over the lifetime of an animal, and it's been shown in squirrel monkey that those cells aren't dead, but they've accumulated the pigment melanin, and that in fact they're still acting as dopaminergic neurons. And so my thought, and again, this is the short version, I'm in danger of going off a long story again, which is on the longer video I posted the other day. There are granules in here in the brain that are in double membrane bound vesicles. So it's just reading a review article by Dr. Luigi Zecca, who's one of the primary people who's published papers in the field of neuromelanin. And so what is this black stuff? It's the thing that causes it to be called substantia nigra. And it turns out that what we understand is that if the dopamine resides in the cytoplasm of the cell here, like it is in red, it's sort of a bad thing. And that is, it's reactive. It forms quinones that are very reactive. And that the cell that uses dopamine stores dopamine in these vesicles, and it's packed with presumably antioxidants, ascorbic acid, if I'm not mistaken, among other things, and that it's not well understood why these things are granular, but that the dopamine is stored in these things and protected from oxidizing to the quinone, which leads to a polymerization cascade. And if it's in the cytoplasm, then it can kind of continue to grow in size as a polymer. And that, oh crap, some things just suddenly made sense. More things. And so my hypothesis when I saw this, and knowing that in old animals, tyrosine hydroxylase enzyme is downregulated. And so I asked myself if tyrosine is normally converted to dopamine by the enzyme tyrosine hydroxylase, and that is the primary way in which dopamine is synthesized in the cell under normal conditions, then, oh, sorry, it's followed by aromatic amino acid decarboxylase, the same enzyme. A two-step process. But tyrosine hydroxylase is the first step in that, from the amino acid, and then the carboxylic acid is removed, and you get this dopamine. So that's thought to be what happens in young animals. And the reason why it was thought that dopaminergic neurons died was because you couldn't put an antibody in here and find tyrosine hydroxylase protein anymore in older animals. And that, again, appears to be not true, that if you look for cells that have the granules of melanin in them, these dark black things, that this cell count doesn't decrease, substantially over the lifetime, at least not more than like 10% kind of thing. And so what that implies is that as a cell gets older, it does not synthesize its own dopamine anymore. And so how does it get it? This is where my sort of light bulb moment was, that the granule of melanin itself was in fact binding and storing dopamine. And that's the hypothesis paper that's in the literature, and that's getting some attention. And so I'm going to follow up some on that. That is, Dr. Zecca's group, I started saying that story and I forgot to finish it. They started doubling down on the idea that it has a protein in its core. He thinks it's there has to be some misfolded beta sheet structure, or something like the tangles that you find in Parkinson's patients. But that makes no sense to me, that you would have a mistangled protein. But their theory, their hypothesis is that the melanin forms to try and protect the cell against damage, and that would fit with that hypothesis, that you get these tangles of protein, and that the melanin is an unnatural sort of thing in that sense. But I don't buy that at all. Having now worked with some dopamine and doing some studies, that it polymerizes. It doesn't need a seed of protein, certainly, and it does it at pretty low concentrations and very quickly, and especially if you add something like adrenaline to it, and it really polymerizes quickly. And so, again, what I'm certain of is that once you have that granule, that it binds free dopamine, and that in fact takes over the function of the synthesis of dopamine in these dopaminergic neurons. So that you don't need tyrosine hydroxylase anymore in an older animal. any dopamine that's in the vicinity of the cell will essentially be attracted to that granule. So I want to follow up on that idea and see what it is. Why do I think melanin is important to this process? And this is a glial cell over here. So I'm probably going to have to erase this and start with a new whiteboard, but it's okay. And so, you might think I'm a little bit nuts, but I think what I'm going to do is switch to talking about phosphorus for a minute. Because phosphate is something kind of unique. So phosphate, phosphorus. Okay. Back up a second because phosphorus is kind of cool. I'm still trying to be elevator talk. Okay, so here's our Earth. Early Earth. It's probably Pangea at the time. And here's a bunch of asteroids, meteorite, asteroid. I don't remember the proper term. Cut me some slack. And the Earth was limited by phosphorus. That's thought to be the case. And so the phosphorus came in oftentimes on meteorites. And in fact, the black rock of Mecca was almost certainly a big meteorite with a whole bunch of phosphorus in it. And so, boom, asteroid, meteorite. Which is it? One of them's got ice in it, one of them doesn't. I'm talking about the rocky one. Is that a meteorite? Oh, shit, I don't remember. Well, anyway, it hits the Earth, it goes boom, and it explodes. Pieces of that thing go all over the place. A big chunk of it's left in Mecca. But phosphorus is spread out all over the world. And phosphorus is extremely oxidizing. That is, it'll grab pretty much any oxygen it can find. And in the early Earth, there wasn't really any O2 to speak of. Not yet, anyway. And so phosphorus was taking its oxygen from water. And we got, for a long time in Earth, there was a lot of intermediates. And so there's a lot more to this story. But what exists today is the phosphate ion. And I'm not drawing it very well on this board with this mouse, but this thing that I tried to draw here with a double bonded oxygen, an oxygen here with a negative charge on one side, an oxygen with another negative charge, an OH group. This is what phosphate looks like much of the time in aqueous solution, and that largely has to do with it being electronegative, and that it had another way of saying that is that it had a lot of electrons that were desirable to the chemicals on the planet at the time, and so it gave up its electrons to the oxygen that was in water, if you want to think about it that way. So phosphate formed early on, and another thing we know around this time, of course, that there were lava spewing out of mountains because of the meteorites, asteroids. So they hit the Earth, they cracked it, they spilled the stuff out, right? You got magma, all the magnesium, divalent metal cations, calcium, all this good stuff, manganese, certainly iron. So what happens? Well, if you put these things together, calcium and phosphate in particular make something we call hydroxyapatite. Okay, and so one of the big missing pieces here to all this story, in my understanding, how the melanins are acting in the brain, rests on the fact that calcium phosphate is magnetic. And that, I mean, if you look at phosphorus and you try to draw the electron groups around it, and if you tried to picture this the traditional way we would think about a double bond in there. Well, this, for one thing, this double bond in phosphorus is not really there. It's here also, and then it's here also, and then it's here. It's everywhere. In fact, it's shared among all four of those oxygens. And what does that mean? Well, that's kind of tricky, because if you think about an oxygen with 3 lone pairs of electrons on it, you'd say that had 4 electron groups, and you'd tell me that that was tetrahedral, right? But what would you tell me for the double bonded oxygen? Well, ideally you would tell me that it had 2 lone pairs and that they were occupying sp2 hybrid orbitals in that oxygen, that there was a third sp2 hybrid orbital that was tying the oxygen to the phosphorus. And you would then tell me that there's an unhybridized p orbital here where the electron would be occupying in order to be part of a double bond with the phosphorus. But this is where things get sticky, because this electron on the sp2 hybridized oxygen is not localized there. In other words, if you can picture the resonance structures of phosphate, and imagine that the negative charges twirling around the ring, if you want to think about it that way, the double bond alternating places, and the hydrogen moving around. This has a lot of implications, it turns out, because all of those oxygens are effectively sp2 hybrid. At least a quarter of the time they are. And so this means that the electron has the possibility potentially of acting aromatically. And the flow is moving. If it's moving, it's a magnetic field. And so the understanding, certainly, that with calcium, it is definitely magnetic. I was just reading a science paper showing the formation of essentially bone grafts by using a magnetic field to create flecks of calcium phosphate. I think was the gist of it. And lots of other things that highlight the interesting properties of magnetic properties of calcium phosphate and other divalent metal phosphates. And so I don't think it's limited to calcium. Certainly iron itself is the thing we generally think of as being magnetic. And when you look at what are called rare earth magnets that include higher molecular, higher atomic mass, transition metals and things in them, the metals that are added to it aren't actually, my understanding is, they're not actually contributing substantially to the magnetic field. They're orienting the iron, and so that its magnetic field is maximized. And so that's why rare earth magnets are used in a lot of applications. But that's not to say that other things aren't magnetic. I mean, oxygen itself is magnetic. And that's something that you figure out if you go through and figure out its molecular orbitals, MO theory. If you go through MO theory, remind yourself what that said back in freshman chemistry, that this is paramagnetic because it has an unpaired electron in it. So the idea that hydroxyapatite, calcium phosphate particles, are magnetic leads me to a lot of interesting hypotheses. And so this may be sort of a little bit wild, and almost certainly some holes in it. If you're watching this, I'd be grateful if you point out some of my holes in it so I can correct my mistakes and strengthen my ideas. I'm going to go ahead and erase this part again and start with trying to picture, whoops, I don't want to do that. Here we go. A bone. Okay, I'll draw a femur or something like that. How about? Oh, geez. Okay, so bone is magnetic, believe it or not. And so teeth. And this really has made me wonder a lot. And so you probably understand that in the bone there's some bone marrow. There's a core of it essentially that's hollow, and that this is in fact where the pluripotent stem cells arise in the body. And this has always struck me as being sort of curious. So pluripotent stem cells are the ones that haven't differentiated. We'd say they haven't become a particular cell type. They are pre-cells. They have their DNAs in a state that it hasn't been modified and moved around and expressed, stuff inside it expressed to cause the DNA expression patterns to change over time. So the thing that strikes me about this is, for one thing, even without any proteins or anything else, even if you just had a little small granule of calcium phosphate, it's magnetic. And that has some implications for what it does with water. For example, the water forms a shell of sorts around this granule, and the delocalized electron, which is part of the magnetic properties of the phosphate, now essentially has access to the shell around the phosphate because this is a delocalized system. And if the electron orbitals around the oxygen can adopt an sp2 configuration and allow for electron delocalization, they do, and they will. And so even before you had bone, which has calcium phosphate and collagen type 1. And so what's collagen type 1? Well, collagen is one of the earliest proteins in evolutionary history. It's got a triple helix structure to it. I don't think I can draw that very well, but essentially 3 strands of amino acid strings, and there's a lot of tyrosine. There's a lot of, oops, I was supposed to say TYR. A lot of tyrosine, a lot of glycine, and a lot of proline. Now, the glycine is the smallest amino acid. My take on this is it's there so that these things can fold up into tight bundles. The proline is similar. It's the only amino acid that's got an internal ring structure to it. And so that causes kinks and prohibits things from normal alpha helices from forming. And so tyrosine is the big player here because tyrosine is the thing that's aromatic and therefore magnetic. That is, any benzene ring is aromatic, and that means it's magnetic. It means it has electron flow through this ring, which creates a magnetic field around it. And so that's key now, thinking that if you just had a granule of calcium phosphate, what things would be drawn to it? Well, pretty much anything that's aromatic. And so what is that? Or, sp2 pi conjugated systems. So that involves a lot of the nucleic acid bases, as well as things like amino acids, especially the aromatic amino acids. And so, well, what? So what? Well, what if you accumulate a bunch of things in here, like catechols, and in fact catecholamines like dopamine, simply because of the magnetic properties of this thing. Right? I'm talking about before there was ever any DNA. Well, you'd get melanin, right? You would polymerize, and you would get a light absorbing melanin. Okay, and then what happens? Because at this time on the Earth, it's thought that there's an awful lot of hydrocarbons, reduced carbon chains, carbon with hydrogen everywhere you can think of it. And that there's a, part of the big mystery is, where did all the phospholipids come from that are making up membranes and cells? That has to start with, well, phosphorylated hydrocarbons. And so I haven't got into this yet, but I need to get my head buried in some of this more literature, some of the terpene phosphates, because I think that's my magic link between phosphorylation and membranes and energy production. But again, any input's welcome on this. So if you got this melanin granule and it started absorbing light, so let me draw that in here. My question then becomes, if you have a photon of light, it hits a granule that's got tons of calcium phosphate around. And where is it? Well, it's in a place that has a lot of oil, which is hydrocarbons. Is this the trigger for phosphorylation of hydrocarbons? Is it start with sugars and, or does it start with the hydrocarbons? Maybe it starts with the hydrocarbons, and then that leads to the oxidized sugars, which then leads to the phosphorylated sugars, all of which accumulate simply because this thing's magnetic to begin with and has a granule pigment that continues to attract more of the same monomer, and so it grows. And if it started to create phospholipids, I mean, it's going to make its own vesicles even. So this is sort of where I'm coming from. Now, let me clear this again and clear this up, because you probably think I'm a little bit Looney Tunes, if I'm going all the way back to evolution. But back to my neuron model, and just sort of quickly, what do I think is happening? And the brief, that's probably what Alzheimer's is too. All of these membranes in here are phosphate. There's phosphate all over the outside of them because they're phospholipids, and they're bridged with waters, essentially. And same with these ones. There's phosphate all around these things. And so you ask yourself, how do these things fuse with each other? If there's so much electrostatic repulsion here on these membranes, and a lot of that has to do with the calcium that goes in between them. And so that part, the electric part, has been known for a long time, ions and attraction, and the idea that the calcium will cause the phosphates to sort of wrap around it, which promotes vesicle formation. That's all well established. The thing that isn't well established is the idea that it's the magnetic properties of it that are causing it to coalesce around melanin. So that is, if you get a melanin, it's equals a big pi conjugated system. That's its big defining feature. Otherwise, you can't make heads or tails of its structure. It's not regularly ordered. People haven't been able to solve it. They just assumed it was junk because it doesn't depolymerize nicely. But it is all a big pi conjugated system. And so what is it going to do in the presence of calcium phosphate, phospholipids? It's going to make its own membrane. It's going to draw its own phospholipid bilayer around it. And so I think that this is really sort of key, that what's happening in humans is that we're getting sort of small granules, and that's what we think of as all of these lipid vesicles that are, when we study animal models and vesicle trafficking, that's what we see, all of these things. But I think they're really sort of seeds of melanin, and in the case of Alzheimer's disease, they are granules, polymers essentially of glutamate and gamma amino butyric acid, which is really the decarboxylized form of glutamate, kind of analogous to L-dopa and dopamine. So, oh crap, I just light bulb moment there too, and I keep going on. And so what I'm suggesting is that what's happening in living things as they get older is that these granules that are forming young are coalescing into larger granules, and that these are what we see in older animals and that we sort of associate with aging pigments. And I think they've been forming all along. It's just that once that granule gets big enough, you stop seeing the smaller things, in part just because the melanin granule has become big enough to store and attract all of the dopamine that this neuron utilizes from that point on. And so this sort of helps explain a lot of what happens in a human as we age in terms of melanin that's produced and melanin that might be released, and in melanin that turns into the hair, and why we start graying after a certain age. And it's probably about at the point when the TH, tyrosine hydroxylase, is downregulated in neuronal tissue, about the time when these granules are substantial enough that, oh God, a lot of things are suddenly clicking too. So this has a lot of implications, because these granules, depending on whether there's metal here, they're going to either be repulsive or attractive, and that attractive or repulsive force is going to turn on instantly everywhere around that vesicle at the same time because it's magnetic and it's a delocalized pi electron system. So this suddenly makes me see vesicle trafficking in a whole new light in terms of why things bump into each other and then they get repelled from things, and why do they seem to magically move around. It's not magic at all. It's pi conjugated systems embedded in vesicles. They're displaying their essentially oxidation state on the surface. And So, as I say, I think this is analogous to Alzheimer's disease is glutamate and GABA, but glutamate, this forms what we call chlorophylls. And so I'm beginning to think that mitochondria and chloroplasts evolved first effectively because they form granules of pigment, and that essentially those granules attracted strings of amino acids, which then led to the formation and evolution of protein enzymes. And I'm going to save that for another day. What is, I think, true though today and is widely unappreciated, and this is also going to be saved for another video, but if you think about DNA, oops, so DNA, here's my double circle DNA, and you understand that it's got a phosphate backbone on it, like all this stuff. What's not widely appreciated is that the stacking in there, the ladder that you're well familiar with, that's a pi stacking. And in other words, this allows for electron delocalization and magnetic behavior. And so DNA is your ultimate in organizing phospholipid vesicles around it. And what's going on is that it sets up a gradient of ions. Everything that's positively charged is trying to get to this buzzing beehive of negative charge, but it's delocalized, and so it's going to form little localized spots where it melts and where it's neutral and where it spits out vesicles again of stuff that came in. And so in my mind, this is DNA doesn't have to encode for anything in order to set up this gradient and start the flow of ions. And I think in the case of melanin and chlorophylls, that in fact what they're doing is they're creating their own proton gradients, and that in fact that flow of protons in the melanin granule is allowing for phosphate, phosphorylation, and energy production. And so those are the details that I'm sort of excited about us learning, and that I'm going to try and follow up on their plausibility in terms of terpenes. But that's the gist of it. I'm going to stop here and see if I can post this and see if anybody gives me any feedback on it. If you do watch it, I'd be grateful if you do that. So thanks for watching.